All right, so thank you everyone for coming back for the afternoon session. We're about ready to begin our afternoon data blitz, same as the morning. Each of the presenters has 3 minutes, and they're going to do a great job with it. Because I took a little extra time with getting this set up, I'm not going to take any more of your time now, and I'm going to hand it straight off to Megan Balk to get us started.
Thank you. So I'm a postdoc here, just moved here in January at the Bio5 Institute. So I'm actually going to talk about research I did at my former postdoc at the Smithsonian. And I'm a paleobiologist. We're a little bit slow to the whole quantitative thing and data science. And so I'm going to talk about my ambitious goals for incorporating data science into our meager data sets that we have in the fossil record. So one of the things that museums already do really well is to archive actual specimens. Some things that they don't do so well is have backup archives. So for example, the National Museum in Brazil recently burned down. And with that were a lot of specimens that can no longer be replaced. They were the only representative of that species. In fact, I just met a woman who was visiting the museum from Brazil who was doing her PhD on a specimen, completely lost it, had to change her entire PhD, and came here to the Smithsonian, which gets to the second point. That is expensive for her to come from Brazil to the Smithsonian, so making data also available to researchers in other countries. And then finally, a way to make use of this data that is just sitting in the shelves. So unless there is active research happening on a particular taxon, on a particular group, that data just sits for decades sometimes. And there could be a treasure trove of cryptic species, of species we haven't identified yet, or of things that I do, which is look at trace fossils essentially on top of fossils. So I study bite marks on marine mammal bones to look at predator-prey relationships in the past during times of changing oceans and how that might affect modern oceans and future oceans. So one thing that the Smithsonian has taken part of that I've contributed 200 specimens to, which I'm very proud of. I realize that that's not thousands, which you guys normally deal with, is to digitize all of these photos. And so I had 2 interns that took pictures of a bunch of marine mammal bones. And kind of the next step that I think that this could really go into, as opposed to just taking care of the archiving problem and making data available to other researchers, is that we can use citizen science and I think machine learning in order to help. And so the things that I want to do is to use citizen science to identify types of bite marks, which I'll be piloting in June, and then also use machine learning to identify unique bones. So either odd trace marks on top of bones, so bite marks, or other pathologies. So there's a lot of vertebra, for example, that are very easy to identify. I think a machine could learn that have weird growths on them, or bones that maybe don't look like other bones. And so these, if you guys have any ideas of how to do this, I'm very new to that field, and I think that paleobiology could really benefit from it. So thank you.
Okay. Hi, everybody. My name is Alexis Work, and I'm a student at the School of Government and Public Policy. And I'm presenting to you today what is essentially my master's research and something that I'd like to continue to build on. So my project is titled Seeking Help, Exploring the Relationship Between the Presence of Non-Governmental Organizations and Reporting of Gender-Based Violence. So the question that I ask is, what factors affect gender-based violence reporting in society? Gender-based violence is a problem across the world. According to the UN, about 1/3 of women experience physical or sexual violence from a male partner or non-partner in their lifetimes. So it's a pervasive issue. And in political science, comparative politics hasn't really addressed gender-based violence in non-conflict settings. So part of my goals in this research is to do that. So the argument that I make in my research and that I'd like to test is that the number of non-governmental organizations that provide services for women in a given area is positively associated with the numbers of reports officially made to report gender-based violence. So my empirical strategy is by using the case of Argentina. I look at month province observations for about 3 years. And there's some endogeneity in my project that I address. So because there's reverse causality, it's entirely possible that in an area, in a community with high levels of gender-based violence reports, that could be representative of historical systemic violence for which NGOs have been created. So in order to address this, I employ an instrumental variable, which is past levels of violence. So in Argentina, there was a military dictatorship in the late 80s or in the late 70s and early 80s. Many people were affected by state-led repression. And as a result, NGOs were founded in many areas that were hit the hardest by the state-led repression. So I use past levels of violence, or the number of detentions in each province, to predict NGOs, or the level of NGOs, thus creating an exogenous shock that allows me to run my model. And thus far, this is still very much a work in progress, and I'm working on running some robustness checks, but my results tend to confirm or support my hypothesis. And I kind of want to address a couple of major implications of that. The first one is that it's incredibly important to have resources, access to services, and awareness about gender-based violence for women, so that they can seek help and access resources. And then in addition to that, scholars of democracy must consider the rights that all people have in democracy, looking at different marginalized populations and how they experience rights. And that's it.
Hello, thank you. So I'm Rebecca Vanderpool. I'm in the College of Medicine as well as the Department of Biomedical Engineering. I'm going to talk a little bit about right ventricular dysfunction in the setting of pulmonary hypertension. So many of you have probably heard about hypertension. Well, I study pulmonary hypertension, which is high blood pressure on the right side of the heart. And these are pressures over 25 millimeters of mercury. You can see here pulmonary angiograms on the left, and then cardiac MRIs on the right. Up at the top are patients without pulmonary hypertension, and at the bottom are patients with pulmonary arterial hypertension. You can see the differences in the size of the right ventricle, as well as in the pulmonary circulation, a loss of the vasculature. There's significant vascular remodeling and the loss of vasculature, and right ventricular failure is the main cause of death in patients with pulmonary hypertension. However, the right ventricle is not a current target for PH therapies. So one of the ways that I help, or what my research does, is we help engineer simple solutions that perform complex analysis on standard clinical data to identify novel therapeutic targets in the right ventricle. So all of the patients undergo right heart catheterization, so they stick a catheter into the right ventricle and measure pressures, and the data gets stored clinically. However, most of the time, clinicians only use a couple data points from this. We can go in, we can extract out the data, we can de-identify it, and get the actual RV pressure waveform. We can then do complex analysis in MATLAB, signal processing, and nonlinear fitting to get a measure of RV function. And what we want to do is integrate the imaging with the hemodynamic data to quantify RV function in pulmonary vascular disease, to identify mechanisms of RV dysfunction, as well as to identify therapeutic targets in the right ventricle. So here we use some dynamic programming to segment the right ventricle and to develop 3D models of the function of the right and the left ventricle. We can also do feature identification in the middle there. We also do segmentation of the pulmonary circulation. So what are the changes in diameter in patients with and without pulmonary hypertension, where you can see models there. We use measures of RV function. And then what we really want to understand is, what is the progression to RV dysfunction? At what point in this progression can we intervene and have therapies? So.
Good afternoon, everybody. My name is Marissa Lopez-Peer, and I am a fourth-year graduate student in the Biomedical Engineering program, and I work in the John Conhelos Laboratory that is housed in physiology. So today I will be discussing how does menopause drive the risk for cardiovascular disease. So we're invested in this research because the leading cause of death for women is heart disease. So I'd like to draw your attention to this graph on the left. Here at the origin, we have the prognosis of heart failure. Then the y-axis is a scoring percentage for disease progression over 12-year follow-up. So we see here that the females that are older than 50 and most likely menopausal have the worst outcomes after they are diagnosed with heart failure. So they're at the most risk for dying compared to their younger females and male counterparts. So currently, the preclinical data that we have in this field is only accounting for 10% of the population. So our lab is trying to account for the 90% and the natural progression into menopause. And this 10% is overoptimized models. Also currently, the menopausal heart does not have recorded the details of the DNA and the proteins that go into this progressive disease of the heart. So we're trying to correct that. And finally, we hope with this knowledge that we can understand more about these proteins and DNA level interactions in order to develop more targeted therapies for women. So, like I said before, our model is special because it recapitulates this natural process of menopause that's seen in humans. So we use a VCD model of menopause in mice. We developed these groups that are listed here as pre, peri, and menopause. They track the age progression that women have over a lifetime. So the first group is women that are 20 to 35. The second group is 35 to around 50. And then the last group is above 50. We then take those proteins, isolate them, and feed them into mass spectrometer. The mass spectrometer quantifies the abundance of these proteins that we then align with a software, Progenesis, and make sure that our proteins that are quantified are aligned to a mouse background. We then are able to do a statistical analysis and create visuals to see how these proteins are changing with our treatment. So our ultimate goal is to understand how the loss of estrogen affects this disease progression. Specifically, if the loss of estrogen is increasing this progression to a diseased heart, or if some of those proteins are inhibiting during menopause this disease state. So if you'd like to know more, I have a presentation later at 3.
Hello, everybody. I'm Lisa Vincent. I'm a French student in geography, and I'm here for 6 months as a visiting student. And I'm here to present my work on soundscapes. So what is a soundscapes? It's not very clear for the moment. We are still trying to create the concept, but basically we want to translate the concept of landscapes from what you see to what you hear. So we are trying to develop an approach to understand and quantify and classify all the sounds that you have in your environment. And we would like to create a method that would automatically recognize and differentiate several types of soundscapes from the very human soundscapes with a lot of human sounds like cars, planes, or trains to more natural soundscapes with a lot of birds and insect sounds. So what we do, I'm working with the spectrum, and what I noticed is that human sounds tends to be in lower frequencies than animal sounds. So for example, here I circled some acoustic events. You have a bird, the slashes, you have insects, the large band. And so what I do is that I cut my spectrum into bins, and I sum the signal power in each frequency bin. So if I have a result that tells me there is a lot of sounds in the lower frequencies, I will say that this is a more human soundscapes. But if I got a result with a lot of sounds in the higher frequencies, it's a more natural soundscape. And what we are going to do next, we want to develop machine learning techniques. So the computer will learn how to recognize new sounds and classify them between human or animal. And this is a very new field of research, but there is a lot of things to do. And I think there is a lot of concrete application. For example, it could really help to develop a better understanding of the impact of human sounds on wildlife. For example, we noticed that some frogs can't find a mate because they can't hear each other. So there is a lot of work to do here. And actually today in the world, there is almost no place anymore where you cannot hear any human sound. And also it could be a very good tool for urban planning and creating spaces and places in cities that would be more comfortable by reducing noise pollution and what humans don't like in sounds. Thank you very much for your attention.
Hi, my name is Maria Sanz. I am a master's student in the GIDP Statistics program. And what I am going to talk today about is about the focus of my master research, that is mixed factor benchmark risk assessment. The main goal of risk assessment is to characterize the adverse response of exposure to a hazardous agent. There are many ways of doing that, and one method that is increasing in popularity is using benchmark risk analytics. How does it work? So if we have, let's see, is the point? Okay. If you have several proportions of adverse response through a range of doses, you can fit a model. And from that model, we can get the benchmark dose, that is the dose that induces a predetermined change in an adverse effect. This predetermined change is called the benchmark response. And we can imagine this like a cutoff point between a high risk that we are not willing to take versus a lower risk that we are fine with it. So we also calculate the confidence interval, and for safety reasons, we are always interested in the low limit of the confidence interval. This has been very well developed in the literature, just to fit one dose, a model with one factor that is continuous. But what has not been really addressed is how to introduce in the model a second factor that is a qualitative variable. So as an example, if we want to study the high risk of, to detect high risk to detect kids with high risk of asthma in the future, and we have data related to skin tests, the allergy skin test, and nitrogen dioxide, we have this data in the Tucson Children Respiratory Study database. We can view this model as you can see in the X axis. We can use the data from the alternaria skin, from the allergy skin test that can be measured as a diameter in millimeters. In the Y-axis, the risk of having asthma. And we can categorize our qualitative variable that is, in this case, is high level of nitrogen dioxide versus low level of nitrogen dioxide. So in this framework, we are going to develop the fundamental statistical theory to fit this secondary variable in the model that is qualitative. And we are going to use Monte Carlo simulations to evaluate how these new methods will perform. Our goal will be to develop risk analytic benchmark methods that can be used by biomedical researchers and risk assessors. Thank you very much. Thank you.
Hi, everyone. I'm Katie Prudick. I'm an assistant professor in the School of Natural Resources and the Environment. Today, I'm going to be talking to you about butterflies and other pollinators. Now, our problem is that pollinators are experiencing a decline in their abundance. That means the number of individuals globally is drastically decreasing. It's happening fast. What does that mean for us? Well, probably about 80% of what you ate for lunch today was the courtesy of that female's efforts. She was carrying plant sperm from one place to another, and she gave you food. Talk about unpaid labor. All right, so we have this problem. They're disappearing. We need data to inform our conservation efforts, right? Have you ever tried to radio track a bee? It's not easy. Okay, so what we're doing, this is a big problem. This is a global problem. So we have a big group of people working on this, and this includes domain scientists who have expertise in pollination, the behavior of these animals, who they are and where they are. We also have data scientists who help bridge the gap between domain information and computer science. And then we have the computer scientists themselves who teach us all about the difference between machine learning and statistical learning, which I'm still trying to learn. So we have this really great combined group of people, and our approach here is to use citizens, everyday people, to help go find where these pollinators are and what they're doing. And that includes both ebutterfly.org, and for those of you with children, I highly recommend iNaturalist, so put that on your phone. It's great citizen science platform. And then from this data that other people are collecting all over the world, we are using machine learning algorithms to help us with identification in particular, so using computer vision to help identify species, because it turns out those are actually two different species of bees. I know, who knew? Yes, when a famous evolutionary biologist was asked about God, you want to know what he replied? He has an inordinate fondness for insects. Remember that, there's a lot of them, and they're very hard to tell apart. So machine learning has been really helpful for that. From that, we're getting information on the specific needs of pollinators and how we can help them. So that includes where they are, what they're doing, and who they're interacting with. And from this, we're getting a great framework for precision conservation management. So we're making management plans based on the pollinators at hand. I think I'm good.
Hi, I'm Kyungmi, and I'm a PhD student in statistics department. Today, I'm going to introduce you my research project called Recommender System with Clustering Guidance. So what is the recommender system? Imagine any situation where users like you interacts with a large catalog of items. And these items are like movies on MovieLens, products on Amazon, TV shows on Netflix, songs from Pandora, or news on Google News. Okay. What matters here is that there are tens of thousands, millions, or billions of items, so that it's impossible for user to go through all the items to find out what you would like. So here's where recommendation is needed. So the recommender system is a system where finds the certain items that the user might like. So it's like predicting your preference. So there have been many like suggestions how to do these recommendations. And today I'm going to introduce one of the most powerful ones called latent factor approach. So these models map both users and the items onto a joint latent space where the user and item interaction can be modeled as a inner product of the user preference factor and the item preference factor. So here's how the story goes. So we have some ratings like history of the users, and then we put that into a matrix form, and then we factorize that matrix, rating matrix, into a low rank two matrices, user preference matrix, and the item preference matrix. And from here, what we're trying to do is we're trying to improve this model by introducing some group effect from users and the items by applying the clustering algorithms. So our ultimate goal is to find better ways to compute these group effects that gives us the smaller prediction error. Okay? And here are some our results so far, and I'm still working on it. So if you have any thoughts or ideas regarding like clustering algorithms or recommender system in general, please come talk to me. I'd love to talk to you. Thank you.
Thank you. Wonderful. Thank you to all the DataBlitz presenters. You were all awesome. And now we have our afternoon keynote, and Bonnie's coming up to introduce her.
So it's my pleasure today to introduce Amanda Borens, who is coming to us from the Critical Path Institute, where she works on global health issues. And one of the things that I love about Amanda is that she actually has an amazing and diverse background in everything from information technology to natural resources, and really a complete picture of many different components. So with that, I'd like to have Amanda come on up. Yeah, that would be better. Put it in my pocket. Okay. Hi, everybody. Sorry for the delay in start there. So as Bonnie said, I'm Amanda Borens. I come from the Critical Path Institute, and I'm really honored to be part of this. I think this is fantastic, what Susan and Bonnie have put together today. And I think if I had had this 25 years ago when I was starting out in my career, it would have been a game changer, because the perspectives of women, even today, are not equally represented in this field. So the more opportunities that you have to talk with other women who are in the same kind of field, take advantage of those opportunities, every single one of them. And as you mature and you grow, make sure that you go back and give back to that community, okay? So I'm gonna start just talking a little bit about Critical Path Institute. We're a nonprofit here based in Tucson, Arizona. We also have offices in London and in Ireland. Oddly enough, we moved from London to Ireland because of Brexit recently. So it's important that because we work with the European Medicines Agency, we have to have a presence in Europe, and London suddenly became not Europe recently, as some of you know. I think something that I want to just take a moment to mention is that what we do is we came about as a result of the FDA's Critical Path Initiative, which basically was the FDA saying it's too hard to get medical devices and drugs approved. It's too hard, it's too bureaucratic. What can we do to make that better? And so Critical Path is an institute that was formed to sort of answer that call. A lot of what we do is working with particularly underserved diseases and underserved populations. So illnesses like muscular dystrophy or tuberculosis. I'm going to talk to you about a couple of examples today, but the thing I want you to keep in mind is just this general concept that most of the pharmaceutical industry innovation happens in the first world, and that is not always ideal, right? Because if there is no profit to be made from selling drugs to people in poor countries, there is no innovation in that disease area. If there's no profit to be made from selling drugs to patients that are very uncommonly affected, there's not really a great profit margin. That's an underserved disease. And so that's something that we specifically really try to target at CPATH. When I figure out how to use the button correctly. Okay, so now my part at CPATH as an architect, as the development manager and the data platform architect, what I do really is sort of coordinating a lot of aspects of data science. My last role before coming to CPATH was literally as a data scientist at a medical diagnostic device company. And what I do now is much more of the collaborative part of data science. And that's something that I think we haven't really talked about as much today, but it's something that you're going to find is a really pivotal part of data science, okay? Because if you cannot communicate your ideas, nobody is going to listen to you. So we work with scientists from universities all around the world. We work really heavily with the FDA in Washington, D.C., and with the EMA, and now in Amsterdam. And I just want to take a quick moment to talk about the actual data science life cycle. So I grabbed this from Berkeley's website. I think when the first time that the phrase data science was used back in the early 2000s, it was sort of a narrow vision of, I would say data science was SQL, Python, and statistics, those three things, right? And it's a much bigger, much more diverse group of people. Sometimes the roles that you see in this data science lifecycle, I've done pretty much all parts of this at some point. Sometimes in one job, I've done all parts of it for myself to get an answer to a question. But I'm in an organization now where we are really highly specialized so that I do a lot of the architecture and then an oversight of other pieces of it. But we have people who specialize in the data management aspects and the data science aspects as far as creating the predictive models. My part really heavily focuses on technology and analytics. I happen to have a master's degree from University of Arizona in natural resources and the environment, go Cats, which has nothing to do with global health, right? But my master's work, and also pretty much all of the work that I've done for the 25 years of my professional career, has had a heavy data component. So I see lots of familiar faces, and the other thing I would advise you all to remember is that it's a small community, and it's especially small if you're a woman, right? So you're gonna run into the same people, you better be nice. So I work in life sciences, and it has a lot of parallels because the work that I did as a scientist really informs the kinds of statistical models and the inferences that I'm willing to extend. And it's something that I see a lot of data scientists lack. So if you can get yourself experience in one of the hard sciences and combine that with statistics, you will be so much more valuable than a person who's a really great programmer that knows statistics very well. Because when you work in the life sciences, you need to be ethical in your extension of inferences, and I've seen that underestimated a lot. So specifically today, I'm going to give you two examples of how do we transform data into information, because that really is the crux of data science. That's what we're trying to do. And today, the problems are very heavily geared toward two things, in my opinion. One is we have so much data that it's no longer helpful. We can't manage it. We can't deal with it. It's inaccessible because there's so much of it. And the other problem is I can't find any examples of this thing in order to make a predictive model, right? So I'm going to give you two case studies that demonstrate that. One is tuberculosis. And I want to start by asking, has anyone in this room had or known someone with tuberculosis? Okay, so of those people with your hand up, how many were in the United States? Those cases were in the United States. So one. And that's why it's important for us to recognize how many people are affected by tuberculosis. Not in the first world, but in heavily in low and middle income countries. This is a huge global crisis. About 10.4 million people in 2015 were affected by TB. 1.8 million of them died in that year. That's a lot of people with an infectious bacterial disease, right? The bigger concern for us in the first world is the spread of bacterial antimicrobial resistance. So multi-drug resistant and extremely drug resistant TB cases are on the rise. About 1 in 4 of those MDR TB cases are detected. So 3 out of 4 never even picked up. And about half of people with MDR TB will die from it. They will not be cured. So it's the leading cause of death among people with HIV. And I think probably the biggest thing that I've learned about working with tuberculosis is that because it's a bacterial infection, the process of identifying your disease, of diagnosing you with tuberculosis, is a culture that takes weeks, literally weeks. Once we've identified that bacterial isolate, we put it through a series of tests with antimicrobial agents to see what will actually be effective. And that whole process takes about 16 weeks. So when you have TB and you go to a clinic and you're in the middle of nowhere in, say, South Africa, you've made it from your village miles away. What really happens in reality is not that they go through a 16-week culture. They throw a drug at you and they hope it works. And so if that drug doesn't work, and it very often doesn't, you're declining quickly. You're infecting everyone in your village. Right? So what we have to get to is a place where we can cut that process of 16 weeks to diagnosis and treatment. We can cut that short, and we're talking about using targeted sequencing, so genomic technology these days. Right now, there's one test on the market, and that will tell you if you are likely to be resistant to the first-line drug that's used in tuberculosis, and that is literally the only thing we know. So what we have to do is identify more of those resistance genotypes. And so some of the data challenge here is now we're entering into the world of whole genome sequencing for bacterial isolates and combining them with the testing that they do in microbiology labs to say which drugs are effective and which are ineffective. It requires terabytes of data to kind of find these relationships in the genome. And one of the things that I find in my work, and one of the things that I find very satisfying, is that I'm able to take that information that epidemiologists and scientists who are the brilliant leaders in their field that have no idea how to handle terabytes of data, I can help make that possible for them. So here's an example of a very simple likelihood ratio of combining the results of drug testing on amikacin, which is cut off here, but amikacin and which genetic mutations were identified in that particular isolate's genome. And what you'll see is that it becomes a big problem because now we're looking just at single nucleotide polymorphism. So we've reduced the problem by putting this through a bioinformatics pipeline. But even when we go to combine all of the possible mutations with all of the possible drug results, you get into hundreds of millions of records, right? So we're working with the World Health Organization now. We've just installed the first global TB surveillance system that's based on genomics in the world at the World Health Organization. And they have the power to receive isolates from member countries in a very systematically designed, randomized trial kind of way. They specifically select patients from specific areas in a randomized design. And this gives them the ability to do surveillance in the past using microbiology labs results, which is often difficult in first world countries, right? So now they're branching into, in this partnership with us, they're branching into using genomic testing as a way of surveilling TB around the world. So what we're expecting to happen is definitely that they'll be tracking resistance genotypes and the rise of that in certain countries. But what we're also hoping will happen is that they combine their past surveillance work in microbiology with the genotypic testing results so that they can help everyone in the world identify which of those mutations are conferring resistance to different drugs. So I throw on some fun pictures here of our data managers who are an important part of all of this work, the cleaning and the management and the quality checks and the loading and the mapping and the building of databases. All of that work happens on the front end. In the bottom there, you'll see our bioinformatician who's actually just finished his postdoc with us, and he's an employee of the CDC now. So we get the privilege of going to work with him at the CDC in Atlanta. And they wouldn't tell me about the place they hide the zombies. So this is a little kind of a visual overview of the surveillance platform that we've built for the WHO. It involves a bioinformatics pipeline on the top left. So all of the genome sequences go in there. We produce for them variant reports. It's all quite automated. Those variant reports get mapped into my SQL database, which is really heavily optimized for large amounts of data with some pretty advanced indexing and annotation for mutations, for example. We combine that with whatever curated phenotypic data they give us for each of the isolates. And then we are working now actively on producing the kinds of reports that can help them sort of get a picture of what it looks like to have TB in Djibouti. And so what we hope is that this could really guide first-line drug recommendations. Because like I said, if you're going to a clinic in the middle of nowhere, you're gonna get the drug that is the most likely to work, right? So if the WHO finds that Djibouti has a huge number of resistance genotypes that are related to resistance to rifampicin, they might change that first-line recommendation to something that is much more likely to work. And hopefully, over time, the more information we gather, the more we can learn to inform those kinds of decisions. So one of the ways that we have helped in this process is to reduce the data burden. So a perfect example is just that you start with a whole genome sequence file, which is 1.5 gigabytes. And by the end of our pipeline, you're down to 1,200 kilobytes, right? So it becomes a much more manageable problem. The second thing that we do is we really translate their actual scientific questions into technology solutions. And that looks like gibberish to somebody who's an epidemiologist, right? But that's our cloud, that's our platform on Amazon Web Services that they're using. Giving them access points and things that are easy to use, like workbench tools for MySQL and open source databases and things that they don't have to worry about sharing with countries. All of those things are helping to enable the work that they do. And then finally, I said this before, communicating the usefulness of the data. It won't matter, all this work that we've done, it won't matter if the people who are making the decisions at the WHO aren't actively using these data, right? So telling the story with reports and analyses has been an important component. And so there's me sitting in the president of the council's chair at the World Health Organization. Matthew, our bioinformatician, he was born in Nigeria. He's a US citizen now, but he was happy to show his support for his home country. I think probably the second kind of scenario that I described to you that is common in data science is a dearth of data, right? So the first one is I'm buried in data and I don't know what to do. The second one is really common in some of these disease areas, and Alzheimer's is a perfect example. One of the reasons that clinical trials are so expensive is that we really don't understand, we humans don't understand the way that Alzheimer's disease progresses. We don't know what the early signs really are. We don't know how far back you can see the difference between I'm forgetful and I have early signs of Alzheimer's disease, right? And because of that, what happens when you go into a clinical trial for a drug? You need to show that you have helped the patient. And how can you show that you have helped the patient if you have no idea what the normal progression of the disease is? So that's been a real challenge. I think you probably have seen headlines about how many drugs have failed. Honestly, we have data that in our database for clinical trials that show us that patients with some of these great new treatments actually do worse. And we know that that's because we haven't teased out the variability and the noise. We know that those patients probably would have been doing worse, right? But there's no measurable way to show that any of the new drugs are effective. So we have a huge amount of variability in the way that disease progresses from one individual to another. And we have really inefficient clinical trial designs. So one of the things that we at CPATH do is we work as a neutral third party, and we say, okay, you're not sharing your data with each other. I'm not gonna get Pfizer to share with Merck, right? But what we do get them to do is share with us their failed clinical trials. And so we keep those data private. Those are in a neutral third-party location. But what we do is produce things like this clinical trial simulator. So this clinical trial simulator is based on the world's first mild cognitive impairment model that was produced by our data science team at CPATH. And this clinical trial simulator allows a pharmaceutical company to go in and say, what would I know that these things will happen? And it gives them a general idea of the population that they should be targeting with their drug. And the reason that that's important is that they are more likely to be able to show an effect if they have accounted for the variability within patients, right? So we have this mild cognitive impairment model. We have this clinical trial simulator, and it came from using, I think, 24 failed clinical trials from pharmaceutical companies around the world. None of them can see each other's data, but we can see all of it, and everyone who shares their data benefit from this clinical trial simulator. So we got a letter of support from the European Medicines Agency. This is the first time one of the regulatory agencies have recommended or supported the use of any sort of disease progression model in Alzheimer's disease. And this came from a lot of political wrangling to get people to share data. So we've talked a lot today about programming languages and what's useful, but I'll tell you that a lot of what it comes down to is getting people to work together, getting people to share their data, understanding that there's no risk. And we see even with academics, we're working with 3 different academic institutions in Alzheimer's now, and we have one partner that literally won't let the data leave their lab. You can look at it, but you can't download it or take it out of their lab. And so I would say that don't underestimate the need for soft skills when you work in data science, right? So why is this particular thing significant? Because data hoarding has been an issue for these competing entities, and sharing data even with academic institutions has been difficult. We don't have huge amounts of longitudinal data. So we have the baseline for a clinical trial simulator, but it absolutely needs more data. It absolutely needs to see what it looks like from childhood all the way through to adulthood. What does Alzheimer's disease look like? And so what is our role there? And in our role, in this particular case, when we're looking for data, is to be a neutral, safe third party for sharing, a go-between between these competing entities. And then at the end of the day, everyone benefits from any sort of regulatory tool that we provide. We're making the clinical trial process easier and cheaper by saving them the trouble of failed trials, right? So I think just in closing, my general thoughts are that data science encompasses many more roles than programming and statistics, many more. I have seen in my career all kinds of strange things happen in my life with opportunities, and I took advantage of every one. I have done inspections of flood control facilities in Pima County when I worked in the riparian habitat group. I have done data munging of just garbage data as part of a project for when I was in graduate school, just doing a side project in EEB. I have done medical device work. I worked for 10 years in healthcare informatics. I've done pretty much anytime somebody said, would you like to try something? I take advantage of that opportunity. I've built a server physically from start to finish just because somebody said, hey, I'm building a server, do you want to help? And I think probably the biggest challenge in data science in my experience is that, that you come out of school thinking that if I just learn these 3 languages and I take that statistics class, I'll be all set. But it is such a deep and complex field. The richer your experience, the better your output will be as a data scientist, without question. And that richness of experience includes standing in front of people talking. It includes selling people on your ideas. And it includes data visualization tools to show people why the number and that p-value is important, right? And remember that a lot of the people who are going to hear the p-value have no idea what a p-value is, right? And then I think probably the other thing I wanted to say today is that when you are here at this conference, you're talking about academic applications of data science, and a lot of it involves science. When you go out looking for a job in industry, if that's something that you end up doing, you'll see lots of jobs for advertising. You'll find things like Facebook ads, Amazon ads, Google ads, Pandora. Everybody wants a data scientist in those places. But there are other opportunities that are not. There are opportunities that are for the good. And for me, data science is such a challenging field in that you can't ever just coast. You're always having to learn new things, right? And the only thing that keeps me going is that I really feel passionately about what I do. I love what I do. I get to help people. If it's overwhelming and I'm tired, I just know that even a little bit of work that I do is gonna help. It's gonna be better for people, right? So just remember that there are options in data science that are not in the typical places that you're gonna see on the job posting websites. And then the last thing I'll say is, that if you have any questions about any of the things that I really quickly skimmed through, or if you have questions about the work we do at CPAC, please feel free to reach out to me anytime. We have internships all the time, and we love to meet new people from U of A. Okay, thank you.
So we have this room, Bonnie Hurwitz talking from Louis Pasteur to CSI. Up in the second floor, S215, Bonnie LaFleur is talking about novel approaches in displaying functional classes of miRNA using compositional data. And on the fifth floor, did anybody go up to the fifth floor this morning? Isn't it beautiful up there? Yeah, you really should go up just to look down. On the fifth floor, standing in today for Erin Leahy is Misty Ring Ramirez, and she's going to be talking about universities' commitment to interdisciplinary research to what end. Great talk from somebody in sociology, so if you want to go check that out, go ahead, and we will meet back here at 2:30 for the next panel discussion. If anybody needs water, there's still some in the back, and there's still, I think, a few more lunches if anybody needs a little bit more food.
righty. Can you guys hear me? Hoo-hoo. If I can get this on. Okay, thanks for sticking with us through the afternoon. Today I am going to really bounce off what Amanda was just talking about, and in using these kind of new next generation sequencing techniques to start to understand the world around us, and in particular the microbes around us, and really start to ask some questions about why are we still using technology from hundreds of years ago to figure out what kinds of microbes we have in this world when we have CSI now. We have these modern sequencing machines that fit on our desktop. So why can't we use them? So as we think about the world around us, it's important to realize that from the moment we're born, we have microbes that are covering us both inside and outside. And these microbes really go across all different domains of life. So they're bacteria, they're fungi, they're archaea. We even have mites and viruses that live on and in us. And this complex ecosystem is really important to our function. And in particular, these microbes have a capacity that we as humans have not even evolved ourselves. So when you think about, the salad that you just ate today, we can thank our microbes for really helping us to digest that plant material and break things down. And we don't have to do all that work. So the question is, as we start to think about, as we go into the clinic and we're sick and we're trying to understand what's wrong, and is it bacterial, is it viral, do I take an antibiotic? It's important to realize that when we send something off to culture, that the vast majority of the microorganisms that live around us don't culture. So 99.6% of the environmental microbes in our world and in our universe, we know nothing about through culture. And although it is better for most clinical viruses, where you maybe get about 50%, there's still a huge amount of unknown that goes into these diagnoses that we see every day. So the question is, as we move into this world of, are these really the microbes we are looking for? And I sometimes wonder, I feel like someone's just putting a little shield over my eyes saying, no, these are not what you're looking for. And they're hiding and they're in there somewhere in the data. So let's see if we can go and find them. So one of the things that we use as a technique is we're using metagenomics. And with metagenomics, the way I like to think of it is sort of like a puzzle. And in fact, it's not just one puzzle, but it's really many puzzles that are thrown together. And my daughter is a great example of this. But what she does is she loves to do puzzles, and she just will pull them down off the shelf. But one of the problems with that is that they tend to all fall down, and then they spill all over the ground. Some go under her bed, the dog eats some, we lose, different pieces of puzzles everywhere. And then you end up with this picture that you're really uncertain as to what all those things are, and you're trying to piece that puzzle back together. Well, microorganisms and the genomes of microorganisms are very similar. And in fact, there are different parts of these microbes that are like blue sky, where we can't tell that one organism is Staph aureus and another is E. coli. And there's really these shared regions of the genome that are similar between these different organisms. So how do we differentiate this? How do we put the puzzle back together and really figure out what we see? So one of the things that we think about oftentimes in the world of metagenomics is that the organisms aren't really there in the community at exact abundances, right? So there's some that are really dominant and are in the community at high abundances, and then other things that are more rare, or the kind of long tail of the microbial world. So some of the picture I can put back together very easily, such as in this case, the Golden Gate Bridge, and other aspects, let's say the Eiffel Tower, I might not have seen. So I really haven't sequenced deep enough to really understand it. So as we think about taking these technologies and converting them into the clinical space and understanding and using those technologies, we have to realize their limitations and what are we looking at and what are the data science problems we have with trying to put all of this information back together. And I think it's also important to realize that there's really many different components to this. There's the good, there's the bad, and then there's the unknown, right? So we have the good, which a lot of people kind of forget to realize, that the vast majority of the bugs that live on and in us are our friends. They are taking up and occupying this niche space all over our skin that prevents those pathogens from coming on in our body and taking over. So we like our bugs. And then in addition to that, there are all kinds of, as I had mentioned, these great microorganisms that are in our gut that really help us to break down the food. And so really quite an important amount of capacity that they offer. And then there's the bad, we know the pathogens, but then many, many others that are just simply unknown to us today, that we are trying to now understand, not by taxonomy or trying to label who these bugs are, but instead by looking at their sequence patterns, just very simply their molecular fingerprints in these different communities of organisms. So I can look at a particular sample and I can know nothing about that microorganism. I don't know anything about where it might exist on the taxonomic tree. But if I can say that that particular microorganism is only found in a particular diseased individual with colorectal cancer, then that's important. Those sequence signatures and those patterns and using this machine learning based approaches to find that is important. So as we think about the good, we should also realize that most of the microbes in our body actually outnumber our own human cells. So thank you, microbes, for being more than me today. And they're also in our environment, right? But let's talk a little bit about the bad. So one of the things that my lab does is we study neutropenic fever. And what neutropenic fever is it's a condition that cancer patients get where they have had their immune system knocked down from chemotherapy, and as a result, they are susceptible to all of those wonderful microbes that we were just talking about in our environment that are normally not pathogens, that normally our own body fights against, but are now suddenly pathogenic. And what's worse is, remember the slide I showed you early on, that 99.6% of these environmental pathogens are just simply, we can't look at them in culture. So what that means is that the vast majority of those bugs don't grow in culture, and we end up telling the patients, we don't know what you have this broad spectrum antibiotic, we hope it's not fungal, good luck. And that's pretty much the way it works, right? So what we're trying to do is we're trying to say, okay, now let's go from this sample collection and really pulling microbes out of blood, which is not easy, mind you, and then isolating the DNA and then sequencing that and trying to understand these novel organisms that exist within these blood samples for neutropenic fever patient samples. One of the things that a lot of people don't realize is that when you take those data and you just do what everybody else does, which is you throw them up against the human genome to remove all of the human reads, because guess what? When you sequence, you get mostly human. That actually, that particular pattern, doing that is probably the worst thing that you can do, because in our own human genome, there are also microorganisms that are embedded in our human genome. So you lose a bunch of your precious microbial sequence as a result. And instead, there are some other methods that we can do to essentially capture those by looking at a more extensive database. And then in addition to that, we also have to ask whether or not someone has an infection. And one of the things that we've realized in our lab, that when you sequence something, you can sequence stuff that's actually not even there. So you are sequencing things that are just floating in from the reagents, and the patient doesn't have a pathogen. They are actually not sick with something, but you're sequencing all the things in the environment. So it's important to realize that when we start to look at these clinical samples, that we have to worry about the contamination, and that when we see things that are really there, they're really there in high abundance. We're seeing those things being really amplified up. And then also important is that we start to look across like how much coverage we have of these different organisms that we find in the blood. And so in this case, these are 3 different patients that we sequenced. One of them had Pseudomonas fluorescens. The other one had a human parvovirus. And then another person had a tortinavirus. And one of the things that we found was this particular tortinavirus, we only saw a very small portion of the genome. And as it turned out, that's an origin of replication. And what we caught in the blood of that patient was the virus actively trying to replicate itself before its entire genome was there. So really started to see these new and novel ways that the viruses are in fact sequencing themselves off our own human genome, because that particular virus is embedded in our genome. So it's really fascinating the types of things we can see. And then lastly, we started to realize that as we start to look across time, we can really understand the different pathogens that we see by their growing abundance over time. So it's really not just these single time points that matter, but really understanding how they change with time. That's important to understand the pathogenesis. So with that, I want to conclude and say that we are still actively learning about all of these different microbes and things that are in our world and in our blood. We explore them through these genomic data sets, but when we look at these genomic data sets, we still have quite a bit of information that we have left to learn. And it's really these machine learning and pattern-based approaches that are helping us understand what's in there. And even worse for viruses, where we have about 95% completely unknown. And so as we move forward, you can imagine that maybe someday we'll end up in the Amazon of the world, where we can take our sample and send it in, and then we can compare it to everything else in the world, and we can say, oh, hey, customers with this particular microbial gut microbiome, we're typically healthy. Awesome, I'm doing a good job. So here's some recommended solutions. And with that, who are you traveling with? All right, thanks. And I'm Betty.
Should I start? I'm just gonna stand.
Although, can you hear me?
Yeah, probably use this one.
Okay. Should I start? Okay. We have 10 minutes, yeah. Okay, good afternoon. We're going to continue. I'm Alethea Ida, an assistant professor in the School of Architecture, and I'm going to speak briefly on data integrated design processes for emerging environmental building technologies. My research basically integrates data from different scales in the built environment. So I look at information from the regional context and urban infrastructure, building systems integration, and then the building integrated technologies. So that includes our building enclosure systems, which are essentially the exterior walls of our buildings to sort of mediate the differences between outdoors and indoors environmentally, down to material scale information. So we usually start with lots of large scale data sets from climate information for the locations in which we're considering building design. The way that we look at and review the climate data varies in terms of the visualizations and graphs. We also like to break that down into temporal conditions for seasonal and diurnal fluctuations, and really starting to kind of address the differentials that might happen between different sets of data in order to it's basically like an entropy guide, which allows us to inform some qualitative aspects of building technology design. So in general, when we visualize climate data, we might look at an annual hourly condition. So these maps on the top that you see essentially can be read from left to right, starting in January, going all the way through December. And then from top to bottom, or bottom to top, depending on the configuration, throughout the hours throughout the day. So as a holistic map, the one on the top is a dry bulb temperature, the one directly below that is relative humidity. We start to kind of see where the concentrations of differences are in thermal gradients, moisture gradients, other aspects of the environment that are significant for building design information. We're also interested in what we call the spatiotemporal information, so the implication of the geometry of solar path and how that influences and drives the thermodynamics of building scale. I take a lot of data into building simulation platforms to basically simulate the energy performance metrics for heating loads and cooling loads, and we look at that through different zones of buildings. So you, with all the little maps that you see on the right, those are depicting different zones of floor plates of a building. So you can imagine if a floor plate had a zone that's sort of in the core, we usually have high cooling loads in those zones because we don't have direct access to the envelope where we can access daylight or airflow or other dynamics from the climate. So at any given time within a building, we actually have different thermodynamic flows occurring. So the entire building does not need cooling all at the same time, the way our current systems predict they do. So we use this data in processing visualizations, active animations, and also in a range of permutations in terms of building program use. So is it a commercial building or a residential use? That influences the human need and the human dynamic on the interior. Then we also look at different building form and orientation and material systems for the enclosure, and that also influences the dynamics. All of this integrated with the large-scale climate data sets gives us tons and tons of data output. And we also integrate that climate data when we start to look at building form analytics in terms of solar radiation mapping on building facades, understanding the intensities of energy, and also the airflow dynamics, for instance, to kind of drive some of the pressure differentials that we might need to incorporate for what we call passive design. So we can incorporate natural ventilation, appropriately design the structural conditions, etc.
So all of that takes us to a point where we say, how can we actually innovate or design new building technology conditions for our local site and region? And this is driven in part by all that data analysis, but also in part by regional material conditions and regional phenomena. So in Arizona, for example, we might have very specific building technologies related to biopolymeric analogy or bioanalytical kind of plant system conditions. If we can understand the sort of natural biorhythms of our plant thermodynamics and material conditions, we can translate some of that information into building technologies. In addition, we consider also looking at environmental remediation in the region and finding natural resource that can help remediate some other environmental condition if we can kind of incentivize an economy for that by upcycling material, maybe byproduct material waste into building technologies. So those are just some examples. We end up with a range of different concepts, and then we continue to integrate data in developing these design concepts. And what the integration of data analytics is really helping, and large-scale data is really helping, I think, for our discipline, is the ability to begin to work with multivalent or multifunctional system design. So rather than having just a static material condition at our building envelope, where we might just have one material that's only doing one thing in terms of rejecting heat or letting in light, we can start to look at the range of environmental parameters that a building technology could integrate, such as daylight control, thermal control, moisture control, all simultaneously. And in part, through design development technique, we've decided to use a new kind of method of proposing physical prototypes integrated with simulation platforms in an environmental test chamber. So the environmental test chamber allows for different prototypes to be inserted into the center. And on either side of the chamber is a volumetric condition where we have sensors collecting data on those environmental flows. We have an input that we can control with a variety of either adding heat, adding air, adding moisture, and then we tend to link our prototypes with some type of actuation and controls. And overall, there's a framework that we've developed for intelligent adaptive control. And in part, this links machine learning processes on the simulation platform as a sort of parallel analysis to the physical prototype that's occurring. And part of this work was accomplished with a collaborative team, including Clayton Morrison from Information Science, Technology, and Arts. And he was integrating the theory for model predictive prior reinforcement learning control, a sort of MPPRL method or methodology. So here's just a baseline example of the chamber where we're using an electropolymeric film that basically allows for light and heat control depending upon the input. And we have sensors on either side of the chamber that are photometric. And they basically feed into an algorithm that defines if one side is bright and the other side is dark, do we want to reduce the opacity, or if we're trying to control heat, do we want to increase the opacity? So things like that. And ultimately, that's also linked back to a building scale simulation engine. So we're getting more data output on the potential of integrating those technologies into building designs and the resultant energy conservation potential. Also, I work with bioresponsive material systems. So with the material integration that are multifunctional materials, how we start to integrate responsiveness in our built environment to human interaction through sensing techniques, machine learning integration, and data analytics, and also, again, having data as an output in terms of potentially like water conservation in this case. So this is a biorhythmic evaporative cooling bench. It's a hydrogel skin that basically releases humidity as people engage with it for natural evaporative cooling. And the top cover is a lyophilized hydrogel, like a foam, so it reabsorbs. It's very hydrophilic, and it reabsorbs moisture. We also collect moisture down at the base. So it's about an 85% efficiency on condensate collect. So it's these types of data integration in the design environment that are really helping to drive some environmental performance metrics as we move forwards. So holistically, it's a wide range of interscalar connections and data analysis at a range of disciplines that we would ultimately want to integrate, including the microbiome. I mean, we just had a nice talk on the microbes, but the microbiome of the built environment is a really important and emerging field for building systems as well. All right, I'm done.
Thank you so much. You know, I was just thinking the last several talks we've heard, I think right now it's easy to kind of feel discouraged about the future, but everything I've seen today gives me so much more hope, and just, this is awesome. And now we are on to our next panel. So in just a few minutes, we'll be starting that. This might be a good time for a bio break if anybody needs to pop out and do that, and then we'll continue with our panel.
We're just waiting on one more panelist. We'll start in just a minute. Hopefully we'll have lots of time to. All right, welcome everybody to the afternoon panel session on how to inspire women in data science. I'm Tina Lee with Cybers. I'm the user engagement officer, and I actually have a fun job of thinking up questions and spurring the conversation today. Because women are perfectly capable of talking and introducing themselves, I'd like each of the panelists to introduce yourself and describe why you do what you do. So. There you go. I'm Bonnie LaFleur. I'm a professor here in Bio5, and I recently came back to academia after being in industry for several years. And prior to that, I was an academician for 15 years. And I did mainly biomarker discovery, and I wanted to go more in the translational side of things, and that's why I went into academia. So I developed a few diagnostic tests and got them through the FDA, and I learned all about that level of the process and getting it into patient level care. But then I missed academia, so now I'm back.
Hi, I'm Hong Cui. I'm associate professor at School Information. My research area is biodiversity informatics. The reason I got into this area is because I need a dissertation topic. And I get into this and I fall in love, and there's plenty of challenges. So it's just keep going.
Okay, I'm Susan, as you know, and the reason that I'm in data science, and I'm sad to not see my boss in the audience because this is my rare opportunity to embarrass him rather than the other way around. But it's not really embarrassing. He had the foresight, I guess, to start the Data Science Institute at the university, and about a year and a half or so ago, we were all told, guess what? We're all going to stop what we're doing and start doing data science, which is cool. And at the same time, like, what are you talking about? So I guess I'm here by accident, but I feel extremely fortunate to have landed here.
Hi, I'm Christina Reamer. I am an ecologist by training, and through the process of my PhD, I realized that in order to leverage large amounts of data and answer the questions that I was interested in, I needed to learn all of the skills that a data scientist would use to clean and analyze my data. And I realized through that process that I also really like to enable other scientists to be able to do these things. And so I want to make their lives easier and be able to help them learn, how to program and how to manage large amounts of data.
Great, you guys are inspiring already. Describe a contribution or an accomplishment in your professional work in which you feel your gender was a factor in. Let's see. I gave them the questions early. This shouldn't be a surprise.
The room. Okay, I'm safe.
So one thing you learn in industry, and especially as a fairly high level person in a company, is the influence that you have to have. And it's predominantly men at that level because it's business development people. So as a statistician, there's a double challenge because number one, statisticians are often renegated in large corporations to just analyze data. So you have to overcome that hurdle. And then the second hurdle is as a woman, and especially as one who's not shy, trying to use it's kind of like you can't be too assertive, even though it's okay for men to be assertive. And so you have to learn a different way of influencing if you actually want your voice to be heard. And that sounds a little more negative than I actually think it is, because I actually think it's a positive thing to be aware and cognizant of that, because you're more effective if you keep that in the back of your mind. So that's probably my proudest moment, was like learning that part of the engagement with other disciplines that are predominant, or other functional groups is what we call it in industry, that are predominantly male.
Wow. Yeah, I just learned something, being inspired. So in my experience, because I'm trained as an information scientist, but my work is all collaborative with biologists. So I think the advantage of my gender gave to me in those collaborative work is that I think most females, you probably agree that we have the tendency to really try to understand the problem before we dive into it. Like a kid is crying, you want to know why exactly he's crying, right? But in my research, I really paid a lot of attention to what real the problem is, because I develop software, I evaluate software, I want my software to be actually useful, help biologists to solve their research problems. So I think the patience that we have to really get into the bottom of the problem before we actually say, here's the solution, was being really, really helpful to me.
Okay. My answer actually has nothing to do with data science, but it does have to do with being a woman. Very first job I got after graduate school, I was a programmer. I liked the job. And then 6 months later, a man got hired to do the same job at a much higher salary. Now, the company did eventually give me a raise, but it made me think, why did that happen? That's not really fair. But to attract him, they offered him a higher salary. So fast forward many, many years, when I first went into management, I saw the same thing happen. And it wasn't really intentional, but it was something that I wasn't going to let stand. I'm like, we have to fix this. We have to get an equal salary for these females that were hired in at a lower rate. So I just want to encourage all of you, if you're starting out or wherever you are, make sure that you know what you're worth and you don't settle for less, because it can happen, and I don't want you to let that happen.
This is the only question that I didn't have a good answer for ahead of time, so I'm going to pass.
Describe a risk you took, your career path that paid off, and what's your evaluation of why that effort? What I'm trying to get by some of these questions is, your unique experience as a woman, often in a STEM field, which is pretty masculine, as we've seen. So what are things that have worked for you in terms of what you've done, how you've done them?
Yeah, it's really funny, because I think that a lot of times there are these cultural indexes, like there's the Briggs-Meyer test and all of those kinds of things that kind of talk about like what kind of a person you are. It just so happens that I'm actually a very masculine, I mean, it's something that a lot of men fit that profile too. But as terms of risk takers, I think there are a lot of women who don't take the same kinds of risks that men do. For example, like leaving an academic job to go out and do something completely different at not a young age, by the way, which could, at the end of the day, I feel that it helped me become a better, I guess I'll say, a data scientist, because I have a much broader experience base now than I did before. But I think that that actually is something that makes us a little bit different, is that a lot of times we have a tendency to think more about what we, like you had said, we have a tendency to think more about how this is going to impact either our family or we think more about that and are a little less I don't want to say assertive, but like we're always wondering, are we going to land on our feet? Even if we've landed on our feet a billion times before, it's like, is this going to be the time that I'm not? And I have to just say, in my, and not in a negative way, I don't always think that men always think that way. I mean, obviously there are some that do, but I believe they're always thought of as the risk takers, and we're often not. But I think that being risk takers is just as beneficial to us, maybe more so currently, because it's more it's more widely recognized now about the importance of having a diverse workforce, and particularly in STEM fields, if that makes sense.
Thank you.
Oh, I guess I have to admit that I'm not a risk-taking person, that kind of personality. But thinking back, anything that I have done that was like my own decision, like somebody has tried to not let me to do that, but I decided to do it. Right. I think one example probably is my first NSF grant proposal. It was 12 years ago that when our school has very, very little few external grants, and nobody encourages grants because we need publications, we need journal articles. So think about, you have this 1% of chance of getting a grant versus you can spend the summer and publish 2 articles. So this was a struggle, but I decided if I don't try, I will never get it. It really benefited me because after the first grant, the following grant was much, much, much more easier. So I guess this could be something to share with you.
I don't know. Everybody wants to hand this mic.
Yeah, exactly.
So I would say that that moment for me was when my boss, even before Data Science Institute existed, he decided to create a position, his deputy director position, and he invited me to apply. And my first reaction was, no, I'm perfectly happy doing bioinformatics, sitting behind my desk and interacting with computers more than people. But I had been doing that for about 17 years. And this little voice said, you know what, you should try something different. You should push yourself, go for it. And I did, and I didn't think I was going to get the job, but I think a big part of that was having done a lot of work on public speaking and being able to get up and give a presentation really confidently. So whatever it takes to get you there, I really encourage you to just take that chance, even though you don't think you're going to get it. You know, why not just go for it?
So when I started my PhD, I was entering an entirely new subdiscipline that I knew nothing about. And then during my dissertation defense, when my PhD advisor was introducing me, he said that one of the things that he liked when I was initially talking to him before I started was that I said, I have no experience with this yet. And that yet was what gave him hope that I would be totally competent to be able to pick up all of the new information from that field. And I think that I've been pretty conscious about trying to embrace that for many years now, because it's very difficult. Change is scary. Not knowing how to do things is really scary. But every single time I take the leap to learn something new or do something completely different, it always ends up being a very satisfying experience. So I've tried really hard to never say no to a good opportunity and to not be afraid of change. And it's okay to be, and someone was talking about this at the panel this morning, but it's okay to not be good at stuff right away. I mean, you're not going to be good at stuff right away. And some of the fulfillment is going from being really terrible at something like coding to becoming a competent practitioner. So
Great. That's actually a good segue into the last question for this part of the discussion, which is, what are some future opportunities that you see for upcoming young scientists in data science fields? So feel free to talk about your own specific domain, or in general. I thought it was Amanda, who's maybe gone already, you know, her talking about CPATH and what she saw out there in the public health, as well as the, she called it the public good sector, was really fascinating. So hopefully you guys can enlighten us further on some other opportunities.
I don't want to steal Susan's thunder, so I'm going to let her talk more about the specifics of Cyverse. But I think that the harnessing large cyber infrastructure, and by harnessing it, I don't just mean using it in making your code run faster, but using it to help develop interpersonal or interdisciplinary science is probably the biggest thing right now. There are tools to share both your code and also build tools for other people. And I think that harnessing things like cyber infrastructure and helping that go like not just using it, but more using it in a way that you can encourage other people to use the things that you're developing. So it's the last part of a pipeline for a data scientist is the communication of what you've done and the insurance that other people can use what you've done. And if you publish a manuscript and you don't actually have runnable, reproducible code, then likely you have missed that opportunity. And places like Cyverse and cyber infrastructure like Cyverse allow us to do that really easily. And I think understanding and knowing how to communicate using that is probably, in my mind, one of the opportunities that we have in the future.
My observation, I think based on the environment, which is way better than like 10 years ago, this conference is one example that it gave you such a great opportunity to networking, to find new talent that you might need for part of your project. I think the opportunity is there. It's just sometimes we hesitate. We are so focused on what the current work requires us. We try to avoid to get into too big a team in fear of time management, commitments, and that kind of concern. But I think the benefit of reaching out and getting into a slightly new field and learn a new methodology and try to use HPC, those are all great things that you could actually jump from this to there.
I actually wasn't going to talk about cyber infrastructure at all. So, but one of the things I loved about Amanda's talk was that she did mention how important our soft skills are. I mean, if we can't communicate effectively and you have to be able to talk to scientists from any domain, that's just so important, because no matter how great your research is and your programming is, if you can't have good communication with your domain scientists, you're not going to be able to help them. And so I just really want to encourage you to never stop learning, never stop growing, working on your personal skills. You know, whatever it is you need to do, find someone who can mentor you, reach out to somebody you've met here today. But it's so important to just keep growing, because if you stop, the world is going to just leapfrog ahead of you in 2 seconds.
So I, at least in my short period of time in science, have noticed that you know, people are increasingly more quickly learning a lot of these sort of data science skills. But I think one of the difficulties is that there are so many things that you can do and can learn, and there's lots of different tools and approaches and things like that, that I did think that it would be unreasonable to expect everyone to have all of those kinds of skills. I mean, some base level should be, you know, part of the required training as we force it into the curriculum at the grad and undergrad level. But I think that going forward, we'll hopefully see a bit more specialization where people are better at certain aspects of sort of the data science pipeline, and then we'll have more collaborative scientific groups, not expecting everyone to know how to do everything, but instead leveraging other people's skill sets along with your own to produce better science.
Great. I personally see an opportunity for women in particular, because I think as we go forward, women are used to juggling, right? We juggle home and work and family and everything. And so I really see that I think the future of data science, the future of science really involves all that juggling, and women, I think, have a good skill at doing that. The last question is really to inspire our audience today on basically what's the best advice you can give them in terms of taking their career from wherever they are now to the next point. So, I mean, that's kind of broad.
Yeah, so I think that there's technical expertise that you obviously you get through your coursework and through your dissertation, and then, as everyone else has already talked about, that soft skills part is not what we always used to call a technical skill, but it is one of these complementary skills that is expected now, because we are working in these cross-functional disciplines. So, and it's not just soft skills like getting up and being able to talk to people, but if you're not interested in that part of soft skills, I think another one is the tool sharing opportunity. So Shiny apps, shiny dashboard, things where you can take what you're building and share it and make it useful for others. That actually is also a soft skill, because you have to understand what people need so that you can write it and make it and build it so that it's useful over and over and over to other people who may not have technical programming skills, if that makes sense.
I wanted to mention that when we actually work really hard to develop our research, try also to balance your life and work. I personally feel like with a very strong, supportive husband and a family, it really made a difference in my career life.
So choose well.
Work with them. I mean, they're all very, very reasonable. You just need to work with them.
That reminds me of Mara this morning and her "Screw You" fund, or whatever she called it. But what I was going to say actually just follows on to what you said very much, which is the importance of taking care of ourselves. We are not going to be our best selves if we don't take care of ourselves, make time for ourselves, get enough sleep, get some exercise, eat well. And I know there are times when that's really hard to do, but you just really have to make yourself priority number one. And that's my best advice.
That's great advice.
I'll take a slightly different tact and say that I think it's really important, especially if you want to do things like data science or research in general, is to find the part of it that motivates you the most and makes you excited to get up in the morning. And that, you know, there are lots of different things that you could do with your time, and you probably should try to minimize the amount of time you're spending on the things that you dread, like meetings, and hopefully make enough time to do the things you actually care about so that you enjoy your life and you don't like get upset on Sunday nights because that means Monday's the next day.
I think we have time to take one or two questions from the audience if there's something anybody has a burning question to ask. Otherwise, yes. Bring you the mic.
So I have knowledge in math, and I have knowledge in somewhat in the field that they want me to apply machine learning to. So from a career perspective, what are some good ways to find collaborators to, especially I'm in industry, and I noticed I'm one of the few people in industry that came to this particular conference. That's not always true, but what is some advice about that?
Does any of the panelists feel they want to tackle that?
So if I'm understanding your question, you're kind of asking, what's the best way to market yourself to be able to, because you are an amalgamation of potentially many. So you've heard today about how the U of A has managed that by having, you know, the coffee, what is it, the coffee and coding, and then you be the one who starts that. I mean, if your company doesn't have that, then you start it, because those kinds of opportunities, if they don't exist, you've heard many people have strategies for that here today of how they make those collaborations, and one of them is these types of opportunities. And if you don't have that, then you start it.
And I don't know if Marnie is still here, but we do have people from off campus coming to Hacky Hour. There's people from Caterpillar who show up. So you are welcome to join the Hacky Hour if that would help you.
Great.
I want to put in another plug for the Twitter science community too. Any sort of topic that you're interested in, you can definitely find people who are active on Twitter who are talking about those sorts of things, and they can definitely give you more resources and materials and possibly even help you out directly with what you're working on. Go Twitter.
Go. Great. Thank you so much, everybody. Really appreciate your contributions to the panel conversation, and thank you, audience, too, for your questions.
Yes, so we are going to now go outside and look at the posters. They will be judged, the graduate student posters. We're going to have 3 prizes, first, second, and third prize. There's a panel of judges who will be looking at the posters. And I also wanted to mention that after the conference, we are going to be emailing you a link to a survey. And to encourage you to fill out that survey, you will have a chance to win a $50 gift card for the U of A bookstore. Am I right? Yeah. So answer the survey and take a chance on winning $50. Go enjoy the beautiful afternoon, look at the posters, talk with the presenters, and then we will come back in here at 350 to announce the winners of the best poster contest, and then we'll head off to happy hour.